Hello everyone. Today we are going to start with Unit 4 of UGC NET JRF Forensic Science, Part 1, in which we will discuss about analysis of ethyl alcohol in beverages, liquors, biological fluids and breath, analysis of methanol and denaturants, as well as illicit liquor. Now some basic information regarding alcohol. IS 3752-2005 is the Indian standard for alcohol, and according to the section 185 of Motor Vehicles Act 1988, a person with blood alcohol content more than 30 mg per 100 ml of blood is liable for punishment. This is the legal limit. And the dry states of India are Gujarat, Bihar, Nagaland, Lakshadweep, Tripura, Mizoram, and some parts of Manipur. So, what are dry states in which the sale of alcohol is totally banned? Now, AOAC, or Association of Official Agricultural Chemists, determine the percentage of alcohol in a beverage by calculating the specific gravity of the liquor samples. Alcohol consists of variety of compounds such as methyl alcohol, ethyl alcohol, isopropyl alcohol, etc. But our area of interest here is ethanol. So ethanol is ethyl alcohol of any strength and purity with the chemical formula C2H5OH. And what does liquor means? Liquor, when ethanol is when additives are added with ethanol. So these additives can be denaturants, which degrades the quality of ethanol, or congeners, which are the added flavors. So what is food poisoning? It is caused by spurious alcoholic preparation. Spurious alcoholic preparation means which involves denaturants, more amount of denaturants, and make it unfit for human consumption. And when the human consume it, it causes poisoning. And illicit liquor. So there are a variety of alcohol, ethanol preparations. The absolute alcohol contains 99.9% of ethanol. Rectified spirit contains 95% ethanol. Perfume spirit contains ethanol with aromatic compounds. Power alcohol is alcohol or ethanol plus 1% kerosene. Biofuel is petroleum plus 5% of ethanol. And neutral spirit, as its name suggests, its pH would be 7. Alcoholic beverage or alcoholic drinks or portable liquor. These are the beverage that contains alcohol in conformity with the Bureau of Indian Standards, and it is fit for human consumption. It may be intoxicating if large amount of alcohol drink is taken. It can cause negative effects on the body. Now, alcoholic beverages, beer contains 2 to 11% of alcohol, wine contain 8 to 15 alcohol percentage, country liquor 12 to 48, whiskey, gin, brandy 42 to 50, and the maximum alcohol percentage is found in rum, which is 40 to 60. Now, a degree proof and alcohol. So 175.25 degree proof equals to 100% of alcohol, and 100% of alcohol is known as pure alcohol. 75.09 degree proof equals to 42.85% of alcohol, which is present in hard liquor. 50 degree proof means 28.53% of alcohol, which is mainly present in country made liquor. Now, what is degree under proof? So if we say 50 degree proof, then 50 degree under proof. 70 degree proof means 30 degree under proof. Now, after fermentation, alcohol can be of two types, distilled and non-distilled. The non-distilled ones are sweet in nature, and The distilled one have more concentration of alcohol. So spirits are the distilled and unsweetened alcoholic beverages which contains alcohol concentration of 20% alcohol by volume, the minimum alcohol percentage. And after fermentation, the process of distillation concentrates the alcohol, which I have already told you, and it also removes additional additives such as congeners or flavors. Now the variety of alcoholic beverages. Dry wine is made by maximum fermentation, which contains 18 to 20% of alcohol. Sweet wine Moscato is made up by incomplete fermentation and it contains 1 gram sugar per 100 ml of alcohol. Sparkling wine, champagne with excess amount of carbon dioxide, and it contains 5 to 10% of alcohol. Beer is made up of barley. Whiskey is made up of wheat, potatoes, molasses, cereals. Brandy of grapes. Rum made up of sugarcane or molasses. Gin made up of berries or added flavors. Champagne, wine with CO2. And fortified spirit, wine with excess alcohol. Example of fortified spirit are port, sherry. Country made liquors, Feni is made up of cashew, Pachwai is made up of rice, Arak made of coconut flour or sugar cane, and toddy made up of toddy plant or palm. The process of formation of alcohol. First, the fermentation of saccharides is done and then mashing at 50 to 60 degrees Celsius. Two enzymes plays vital role in the process of formation of alcohol, that are maltase and zymase. Maltase converts saccharides into maltose and glucose, and zymase converts glucose into ethyl alcohol. Now, fermented liquor. It can be of two types made up. First is made up of incomplete fermentation. With incomplete fermentation, we'll have liquor with sugar, as the whole amount of sugar is not converted into ethyl alcohol. So the incomplete fermented liquor will contain high amount of sugar, whereas the complete fermented liquor will have high percentage of alcohol, not sugar in it. Now, after the formation of liquor, it can be incomplete fermented liquor or complete fermented liquor. Also, it can be distilled and non-distilled. The distilled one will have high percentage of alcohol, and the non-distilled one will have alcohol percentage under 50%. The steps involved in the formation of beer are malting, that is steeping, germinating and drying the grain. Next is mashing, then brewing. Brewing means steeping starch source in water. Then fermentation. Fermentation of sweet liquid is done using yeast. Then maturing. Maturing is secondary fermentation, and then finishing. Pharmacokinetics of alcohol. The first step is absorption. So alcohol is absorbed via mouth, stomach lining, intestine, and get absorbed into the bloodstream. So small amount of alcohol is absorbed through the mouth, and then with the stomach lining, and most of the alcohol is absorbed through upper part of the intestine. And during absorption, the blood first gets into the arteries. So the arterial alcohol concentration is high as compared to the venous blood alcohol concentration. But after this stage, there is an equilibrium in which both arterial and venous blood have equal alcohol content. Absorption depends on nature of the drink, what alcoholic percentage is there in the drink, Rate and speed of drinking, how much have you drunk, and whether you are drinking on an empty stomach or a full stomach. In case there is food present in the stomach, so the pyloric valve that contains stomach to intestine remains closed, and maximum alcohol absorption is via stomach lining, and it is a slow process. So if the stomach is empty, more absorption of alcohol will take place through small intestine. Distribution of alcohol after absorption, it gets distributed to every part of the body. The distribution of alcohol depends upon the water content of particular part. The more amount of water is present in a particular part, the more alcohol will get absorbed in it. The maximum amount of water is present in brain tissues, that is 85% water. So highest amount of alcohol will get distributed in the brain. Now the last step is elimination of alcohol. So 90% of alcohol is metabolized through liver, where alcohol is converted into acetaldehyde, and then acetaldehyde is converted into acetic acid, and then into carbon dioxide and water. We'll discuss this process in detail later. And the remaining percentage of alcohol is eliminated by excretion, respiration and perspiration. Excretion urine, respiration breath, and perspiration sweat, in the form of urine, breath and sweat. So estimation of alcohol can be done using blood, urine, sweat and breath. All these contains alcohol. The most widely accepted samples for alcohol identification are blood and urine, as they are easy to collect, and percentage of alcohol is identified. Metabolism of alcohol. So alcohol is metabolized through two different pathways, oxidative and non-oxidative. Oxidative is in the presence of oxygen, non-oxidative is in the absence of oxygen. So maximum metabolism is done by oxidative pathway. Alcohol is converted to acetaldehyde by two enzymes, catalase and alcohol dehydrogenase, and acetaldehyde is further converted into acetic acid using acetaldehyde dehydrogenase enzyme, and this acetic acid or acetate is converted to carbon dioxide and water and circulated in the bloodstream. In case of chronic drinkers, the CYP2E1 enzyme plays role. The excess amount of alcohol is converted into formaldehyde, not acetaldehyde, formaldehyde. Uh, so this is the major process that is taking place. This process is found in chronic alcohol, and small amount of alcohol goes in the non-oxidative pathway, gets attached with the fatty acid, phosphatidylcholine, glucuronic acid and sulfate to form FAE, fatty acid ethyl alcohol, PETH, glucuronic acid. With glucuronic acid it forms ethyl glucuronate, and with sulfate it forms ethyl sulfate. Continuing with the metabolism of alcohol, alcohol dehydrogenase removes hydrogen from alcohol and converts it into acetaldehyde. Cytochrome P452E1 or CYP2E1, that is present in chronic drinkers to convert ethyl alcohol to formaldehyde, is this enzyme is present in microsomes of cytochrome. Catalase enzyme is found in peroxisomes, and it helps in the conversion of alcohol to acetaldehyde. Acetaldehyde dehydrogenase is present in mitochondria, and it converts acetaldehyde to acetic acid, And alcohol can also be formed after death by the fermentation of saccharides present in the body with the action of bacteria or yeast. So, how do we differentiate between ante-mortem and post-mortem alcohol? In post-mortem alcohol, ethyl glucuronide or ethyl sulfate is absent. So, these two helps in the identification of whether the alcohol is ante-mortem or post-mortem. What are the changes in the body after alcohol consumption? So, when the blood alcohol content or BAC is between 0.05 to 0.1%, it is the stage of excitement. When BAC is between 0.1 to 0.3, it is the stage of incoordination or confusion. BAC above 0.3 is the stage of narcosis. The fatal dose of absolute alcohol, that is 99.9% alcohol, is 150 to 250 ml in adult and 60 ml in children below the age of 12 years. The fatal period is 12 to 24 hours for absolute alcohol. Now, here absolute alcohol, the fatal dose and fatal period is for absolute alcohol, 99.9% alcohol, not for alcoholic beverages. Then delirium tremens, it is the stage of mental confusion and emotional disruption. Korsakoff psychosis, in which there is disorientation, hallucination and multiple neuritis. So Henry's gas law application in alcohol is the ratio of concentration of alcohol in the blood and breath is constant at a given temperature, and if the temperature is raised, the equilibrium will shift towards the concentration in the air or the breath. So the average rate of alcohol and elimination in breath per hour is 6.5 microgram per deciliter. The volume of alcohol to get drunk, for males it is weight into blood alcohol content by 3.78, whereas for females it is weight into blood alcohol content by 4.67. Volume of alcohol to get drunk depends on various other factors also. The fat cells present in the body, or whether you are drinking on an empty stomach or a full stomach. So there are many factors related to this particular thing. As after absorption, the blood gets into the arteries first. So the arterial blood is ideal for sampling as it is most closely related to the brain alcohol content. Proof spirit is used to denote an alcohol of approximately 57% by volume, which, when poured on gunpowder, burns the gunpowder in spite of it having 42.9% of water content. The lethal dose of alcohol is 0.4 to 0.5% and BAC 0.03%, that is 30 mg per 100 ml of blood. Above it, it is prone to accidents and liable to the punishment. Average rate of alcohol removed is 0.015 blood alcohol content per hour in breath, urine, sweat, feces, milk and saliva. Ethanol is also known as grain alcohol and it is formed by the fermentation of yeast, whereas methanol is also known as wood alcohol. Methanol is not directly poisonous, but its metabolites are. So methanol metabolizes in the liver to form formaldehyde and then formic acid. These metabolites are poisonous. The statement of Henry's law: when a volatile chemical, in our case it is alcohol, is dissolved in a liquid, blood, and is brought to equilibrium with air or alveolar breath, There is a fixed ratio between the concentration of the volatile compound alcohol in air and its concentration in the liquid, and this ratio is constant for a given temperature. I have already explained Henry's law in the previous slide. The temperature at which breath leaves the mouth is normally 34 degrees Celsius. So this 34 degrees Celsius is the constant temperature. Okay, and at this temperature, the ratio of the volatile compound alcohol in air and blood. So the ratio of alcohol in the blood to alcohol in alveolar air is approximately 2100 to 1, or 1 milliliter of blood will contain equal amount of alcohol as that of 2100 milliliters of alveolar breath. So Henry's law becomes the basis for relating breath alcohol to blood alcohol concentration. In death due to alcohol poisoning, blood for chemical analysis is preferably collected from axillary or femoral vein, and alcohol is a hepatotoxic poison. Hepato means liver, so it is a toxic poison which affects the liver. The ethanol concentration ratio of saliva and blood equilibrates after 30 minutes of cessation of drinking or stopping of drinking under normal conditions. World Anti-Doping Agency guidelines to specify the procedure for breath alcohol testing is WADA 2006B, and nandrolone is used by athletes for doping. It is a different thing. The breath alcohol testing is done via WADA 2006B. Now the techniques for identification of alcohol, whether a person has consumed alcohol or not. The very first are the field sobriety test. National Highway Traffic Safety Administration has regulated 3 standard field sobriety tests, which are horizontal gaze nystagmus, walk and turn, and one leg stand, to ascertain the degree of the suspect's physical impairment. Next is the breath analyzer, in which a suspect blows air or breath into the analyzer, and the acidified potassium dichromate, which is initially yellow in color, will oxidize alcohol to acetic acid and get reduced to form chromium sulfate, which is green in color, and the catalyst used is silver nitrate. It is also used to measure the alcohol concentration present in 1/40 of a milliliter of blood. In case of other samples for alcohol identification, such as blood, anticoagulant is added, which is EDTA or potassium oxalate, to prevent clotting. Preservative such as sodium fluoride is added, and it inhibits the growth of microorganisms that are capable of destroying alcohol. So, in case of blood, we use EDTA or potassium oxalate, sodium fluoride. Iodoform test to the sample, Lugol's iodine solution, that is potassium iodide plus iodine, is added along with sodium hydroxide, and it is warmed. Again, iodine solution is added, and then NaOH is added from the side of the test tube. There is a cloudy appearance. This whole mixture is warmed in water bath and waited to form yellow PPT. This yellow PPT is observed under microscope and it contains hexagonal iodoform crystals. So it gives positive results for alcohol as well as for acetaldehyde. The reaction is given in the right side. The oxidation by NaOI formed with the reaction of iodine with NaOH and the substitution of hydrogen by the reaction with iodine in the presence of OH negative and then breaking of CC bond by OH negative ions. Here H2 is removed and double bond is formed in between C and O. In the next step, the 3 hydrogens are replaced by 3 iodine, and in the last step, iodoform is formed along with RC double bond O, RCOOR. Another test is Sulfo Molybdic Acid Test. The reagent is 1 gram molybdic acid in 25 ml of concentrated sulfuric acid. 2 ml of sample is taken and 2 ml of hot reagent is added. Deep blue ring is formed. In case of ethanol, this test shows negative results with acetone, acetaldehyde and dilute solutions of methanol. If the methanol solution is very concentrated, the result is blue color and not deep blue color or dark blue. Dichromate test, which is also used in breath analyzer, sample with 0.2% potassium dichromate solution and 1 ml concentrated sulfuric acid. Greenish blue color is observed. So the ethanol is oxidized to acetic acid and potassium dichromate is reduced to chromium sulfate in the presence of sulfuric acid. So greenish blue color of chromium sulfate is observed. Determination of ethyl alcohol in blood and urine as per Widmark's formula. For blood, A is equals to CPR, where A is the total amount of alcohol absorbed in the body. C is the concentration of alcohol in blood. P is the weight of person and R is constant, 0.68 for men and 0.5 for women. And for urine, the formula is A is equals to 3 by 4 QPR. Here, Q is the concentration of alcohol in urine. Kozelka and Hein or Cavit method. The principle of this method is alcohol easily gets oxidized to acetic acid in the presence of an oxidizing agent. Breath analyzer works on the same principle. In this method, Cavit method, each ml of N by 20 potassium dichromate solution that is reduced is equal to 0.575 milligram of alcohol. Modified Kozelka and Hein method. It consists of 4 tubes which are connected to each other. Uh, the dimension of the tubes is given in the slide. The second, third and fourth tube is kept inside the water bath. The first tube contains 5 ml of 2% potassium dichromate in concentrated sulfuric acid. The second tube contains blood or urine sample with 2 ml of 10% sodium tungstate and 0.5 ml of 2 normal sulfuric acid. The third tube contains 5 ml of saturated mercuric chloride and 5 ml of saturated sodium hydroxide. The yellow PPT of hydrated mercuric oxide traps the acetone and acetaldehyde. The fourth tube contains 10 ml of 0.1 normal potassium dichromate and 10 ml of concentrated sulfuric acid. After 1 hour, the fourth tube solution is taken out and the volume of this solution is made up to 100 ml using distilled water. This solution is titrated with 0.1 normal sodium thiosulfate solution. Now, the 1 ml of this potassium dichromate solution is equals to 1.15 mg of ethanol. The blood alcohol concentration is given by the formula 1.15 into X minus Y into 100 by V, where X is the volume of 0.1 N sodium thiosulfate required in blank experiment, Y is the volume of 0.1 normal sodium thiosulfate required in sample, V is the volume of blood taken for the experiment. For confirmatory analysis of alcohol, GC, gas chromatography, enzymatic method, intoxilizers are used. In enzymatic method, the coenzyme NAD reacts with alcohol to convert the alcohol into acetaldehyde and get converted into NADH. This whole reaction is monitored at 340 nanometer under UV visible spectrophotometry. Denatured spirit. What is denatured spirit? In which a denaturant is added. When the denaturant is added to the liquor, it is unfit for human consumption. Denaturant is the substance added in the denatured spirit. The example of denaturants are methanol, acetaldehyde, chloral hydrate, etc. And a trichloroethanol is an active metabolite of chloral hydrate. Chloral hydrate plus alcohol is known as knockout drops or Mackay fin. Fermentation products, the denaturants present in the fermentation products are acetic acid, methanol, acetone, aldehyde, volatile and non-volatile acids. The additives present in fermentation products are esters, minerals, sugars, flavoring agent, coloring agent and fusion oil. Types of denaturants: completely denatured, specially denatured and ordinary denatured. So in completely denatured, it is the denaturants are pyridine and caoutchouc which is 0.5 liter per 100 liter of alcohol. Also methanol 0.5 liter in 100 liter of alcohol. Specially denatured ones are methyl alcohol plus benzene 2 liter each per 100 liter of alcohol, acetone 2 liter each 100 liter of alcohol, 2 liter of acetone per 100 liter of alcohol, ether, chloroform, turpentine oil. All these are 2 liter per 100 liter of alcohol. Ordinary denaturants are methyl alcohol plus caoutchouc 0.5 liter per 100 liter of alcohol. Next is crotonaldehyde plus pyridine plus methylene blue 0.6 liter in the ratio 0.6 liter is to 0.5 liter is to 0.001 kilogram per 100 liter of alcohol. Now the comparison of methanol with ethanol. So the properties, the molecular formula for ethanol is CH3 CH2OH or C2H5OH and methyl alcohol is CH3OH. The common names of ethyl alcohol is methyl carbinol or grain alcohol, and of methanol is carbinol, Lichtenstein, wood spirit, wood naphthol or wood alcohol. The effect of ethanol is depressant. and the effect of methanol is blindness. Ethanol is produced by fermentation. Methanol is produced by distillation. The boiling point of ethanol is 78.4 Kelvin and as that of methanol is 64.7 Kelvin. The density of ethanol is 0.789 grams per centimeter cube, whereas that of methanol is 0.7918 gram per centimeter cube. The antidote of ethanol is fomepizole or 4-methylpyrazole and vitamin B. The antidotes for methanol is ethanol and fomepizole. The metabolites of ethanol are acetaldehyde and acetic acid. The metabolites for methanol are formaldehyde or formate. The compounds used for the withdrawal of ethanol are disulfiram, citrate calcium carbamide, metronidazole, nitroprazole, and methyl tetrazolethol. The fatal dose of absolute alcohol in adults is 200 to 500, and for children below the age of 12 years, it is more than 50 ml. The fatal dose of methanol is more than 30 ml. 6 to 10 ml of methanol can cause blindness. The metabolism of methanol methanol metabolism is under the liver. Methyl alcohol is converted into formaldehyde, then formic acid, then folic acid, folinic acid, and then into carbon dioxide and water. Here the formaldehyde and formic acid are the most toxic components. Chromotropic acid test for methanol. Sample 0.5 ml plus 5% potassium permanganate, 0.2 ml. After 5 minutes, so saturated solution of sodium bisulfite or oxalic acid is added drop by drop to make the solution colorless. To the resultant solution, 0.5 ml of freshly prepared chromotropic acid is added and the solution is heated on water bath at 60 degrees Celsius for 30 minutes. After 30 minutes, the solution is cooled and violet ring is observed. Schiff's reagent test for methanol. 4.5 ml of solution is taken and 3% potassium permanganate solution 2 ml is added, followed by 2 ml phosphoric acid, and it is left for 10 minutes. Then 1 ml oxalic acid followed by 1 ml of concentrated sulfuric acid is added and the resultant is cooled at room temperature. After that, 5 ml of Schiff's reagent is added and the mixture is kept undisturbed for half an hour. Purple color is observed. Now, tests for acetaldehyde and formaldehyde. Schiff reagent test. To the purple color, concentrated sulfuric acid is added. If the color disappears, it indicates acetaldehyde, and if the color retains, it indicates formaldehyde. Other tests for acetaldehyde is iodoform test, and for formaldehyde is chromotropic acid test. Illicit liquor or illegal liquor, in which the method of distillation is poor, so it contains injurious materials. It do not follow standard procedure. It is made by unauthorized distillery. Adulteration is there. Any liquor manufactured, stored, distributed, sold on contravention of provisions of Central Excise Act, or any liquor on which the appropriate custom duty has not been paid. Any of these conditions will follow. and the liquor is termed as illicit liquor. Thank you for watching. For more such videos, subscribe my YouTube channel Vidhi Vigyan.